Silence, please. Good morning, everybody. It is a pleasure for me today to introduce you to Maribel García-Ibáñez. She got one of the fellowships, the postdoctoral fellowships from Severo Ochoa that were offered recently, and then she started the contract in December. She was born in a little town in Castelló, so you can talk to her in Catalan. But then she studied the degree of marine sciences in Vigo, okay? And it's in Vigo as well. Well, she finished in 2011, and then in Vigo she also did the PhD, supervised by Fiz Pérez. She worked in this group. They are experts in the CO2 system in seawater. Okay, and then she finished the PhD in 2015. And the PhD was on the understanding the anthropogenic perturbation in the inorganic carbon cycle in the North Atlantic Ocean. Then after the PhD, she was working also in the University of East Anglia, also in the University of Delaware, in the National Institute of Standards and Technology in USA, and in Norway in UNI Research. And then Maribel is a member of the Scientific Steering Committee panel on the International Ocean Carbon Coordination Project, and also collaborate with the Ocean Carbonate System Intercomparison Forum. Okay. Maribel she has a strong expertise on the sequestration of CO2, of anthropogenic CO2 in the oceans, and also in the ocean acidification that takes place because of that. And today she's going to make a bit of a summary of her results, particularly in the North Atlantic Ocean. But she also will tell us about the undergoing projects in the Southern Ocean and also in the Mediterranean, which is the focus of the Severo Ochoa Fellowship that she's taking since December. Okay, so the floor is yours.
Thank you, Carles. And thank you everyone for coming to see my talk and for the opportunity to present my results here in the Friday talks and for present myself to the ICM, because most of you maybe don't know who I am. And as Carles was explaining during this talk, I'm going to talk a bit of what I did before coming to ICM, which was, as Carles said, related to water mass circulation and ocean acidification in high latitude oceans, both the North Atlantic and the Southern Ocean. And at the end of the presentation, I will give a slight introduction of my project that I'm doing here that is based on the Mediterranean Sea. So why did I decide to study what I did during my PhD and my postdocs is linked to the fact that the global ocean has mediated the atmospheric carbon dioxide increase because it has taken up to 30% of the emissions since Industrial Revolution. And as you see, I'm going to point here because if not, people on the internet won't know what I'm talking about. But as you see here, this anthropogenic or human-derived carbon enters the surface ocean through air-sea gas exchange. And most of this carbon, as it's represented in this diagram here, is stored in the first meters of the water column. As you see, this is a schematic profile of the concentration. And as we move deep into the interior ocean, less and less anthropogenic carbon can be found. And this is linked to the fact that this, as I said, the anthropogenic carbon enters surface ocean through this air-sea gas exchange. And the limiting factor of this deep penetration in the water column is this represented here by this overturning cell, which is all the mechanisms that transport these upper waters to the bottom of the ocean, which are the same mechanisms that control the ventilation of the deep ocean. And that is why the high latitude oceans are the regions where most of the anthropogenic and carbon in general is stored in the global oceans. And I try to illustrate this in these two schematics here. So on the top one, you can see a schematic of the water mass formation and circulation in the global ocean. So you here have three sections in the Indian, Atlantic Ocean, and Pacific Ocean, and they are all interconnected through the Southern Ocean. And if we focus to in the North Atlantic, we here see the formation of the North Atlantic deep water, which represents 40% of the deep waters in the global ocean, and the other 60% of the deep waters in the global ocean are formed in the Southern Ocean. You here see these 3 blue arrows. They are the 3 formation regions of the Antarctic bottom water. And you can see how linked to this deep penetration of waters, we have deep penetration of anthropogenic carbon signal. On the graph on the bottom part, you see the representation of the anthropogenic carbon signal. And as I was explaining earlier, most of it is stored in the upper 200 meters of the water column, and we only see deeper penetrations of this anthropogenic carbon signal around the Southern Ocean here and in the North Atlantic, linked to this formation of water masses around the Southern Ocean and the subpolar North Atlantic. And this is one of the aspects of my work, studying how this circulation of water masses affects the storage of carbon. But also, I studied how this storage of carbon comes with kind of a cost, with the service that the ocean gives us, and that cost is known as ocean acidification. And I tried to illustrate this with this kind of schematics of the chemistry of the carbonate system. And as you see here, the carbon dioxide is a special molecule because it's different from other gases like oxygen that only dissolve in water. In this case, carbon dioxide reacts with the seawater and forms a weak acid that is not represented in the system because it's so weak that dissociates really fast into bicarbonate ions and carbonate ions. And you see every time we move to the right of these equations of reaction chain, we have a release of protons here. Every time we release a proton, we reduce the pH of seawater. And as you see here, there are double arrows, so it means that the reaction can go to the right and can go to the left. So there is an equilibrium, which is known as a buffer system. And this has kind of 2 consequences, meaning the buffer makes that the pH doesn't change as fast as it should be if we only have one direction in the reaction. But the other consequence is that because the reaction can go to the left, most of these carbonate ions try to take these extra protons and form back these bicarbonate. So in the present oceans, most of the dissolved inorganic carbon, which is the sum of this carbon dioxide, this bicarbonate and carbonate ions, is in the form of bicarbonate. So every time we add more CO2 to the water, we reduce pH, but we also reduce the concentration to this carbonate ion. And why do we care that we reduce this carbonate ion? It has 2 consequences as well. One is that if there is less carbonate ion, there is less availability of this carbonate ion for organisms that use it to form the calcareous structures. Like I just shown you some examples here, but there are many more. And they are in different trophic levels in our systems. But also, as illustrated here in this top graph, if we move from our pre-industrial oceans to the oceans that may be predicted for the end of the century, we have less of this carbonate ion here. It will affect our organisms, our ecosystem, but it would also affect the chemistry of the water itself, because this carbonate ion is part of a parameter that is known as alkalinity. And basically, the alkalinity of the seawater is like an antiacid, as I was saying. The carbonate is part of this alkalinity system. The more alkaline the water, the more carbon dioxide it can store, and the less its pH will change. So we want waters that have a lot of alkalinity to store more carbon and to have less of these ocean acidification effects, right? So that's kind of the basics of what you need to have in mind when I'm going to present the rest of my talk. So now focusing on the studies during my PhD, these were related to the subpolar North Atlantic, which is the region that is represented in this map here. And why we decided to study this region is because it's a key region for circulation, where we have what is known as the Atlantic Meridional Overturning Circulation, which I will explain what this is. And this Atlantic Meridional Overturning Circulation starts briefly with these central waters here coming from the subtropics to the subpolar North Atlantic, which is the region that is represented in this map. And when these waters arrive to the North Atlantic and subpolar North Atlantic, the interaction with the atmosphere cools down these waters and transforms them into what is known as subpolar mud waters. And the end result of these subpolar mud waters and the dense air variety of them is known as the Labrador Sea Water. And this Labrador Sea Water is formed in the Erminger Basin and the Labrador Sea through deep winter convection. And once this water mass is formed, it joins the deep western boundary current, which is represented here in the dark blue arrow, and where it flows over the overflow waters, the deep waters that come from the Arctic and Nordic seas, which are represented here as the Denmark Strait Overflow Water, or DSOW, and the Iceland-Scotland Overflow Water, or ISOW. So this circulation of water masses, where we have surface waters that then sink and come back as relatively new deep waters, ventilates the deeper ocean and also brings lots of carbonate ions or excess ions to the deep ocean. And that created favorable conditions for the growth of cold water corals. And these corals are maybe for you some kind of unknown, because most of our research in climate change is focused on tropical corals. But these corals here, they can live, as you see here are represented, the dots are the locations where we know there are cold water coral reefs in the global ocean, and the color represents the depth where they live. So you can see they can live up to 2,500 meters and deeper in the water column. So they don't have this symbiotic algae inside of them, they are not colorful, and maybe they don't have as much attention, but they are really important ecosystems in the global ocean. And as you can see here, the North Atlantic has lots of reefs, and this is because of this renewal of the waters that happens in the subpolar North Atlantic and in the Arctic. And for a concept that I know some people maybe are not familiar with, aragonite, calcium carbonate, and all this, I want to give you some brief concepts so you understand the rest of the talk. So these corals, as all corals, form their skeletons with calcium carbonate, and the variety that they form is known as aragonite. And it is seen that over 90% of the locations, these locations here, are located in a depth that is shallower than the pre-industrial aragonite saturation horizon. And what is that? So this aragonite saturation horizon is the depth at which the aragonite saturation state is equal to 1. And this is a lot of concepts that sounds like Chinese to anyone, but basically what it's telling us, this aragonite saturation state is represented by omega, and is more or less approximated to the ratio between the concentration of calcium carbonate that we have in situ and the concentration of calcium carbonate that will be at saturation, that the maximum kind of, not maximum, but the thermodynamically maximum of concentration. So if we have in situ higher concentration than saturation, we have a supersaturated water. The ratio will be over 1. And if we have in situ a concentration that is lower than the saturation concentration, we have unsaturated waters, and the omega will be below 1. What happens when we are in undersaturation conditions? It means that thermodynamically, aragonite tends to dissolve. So by just putting this aragonite on this kind of water, it will dissolve, the structure will dissolve. And that is why we care about this in the corals. So in these pictures here on the left, you have a cold water coral, as I said, it's not colorful, living in a supersaturated water with omega of 1.1. It's just slightly supersaturated, but it's still supersaturated. You can see here we have a 3D structure, right? It's like a reef as we are used to see. And as I said, they are not colorful, but we know it's alive because we see these white parts here. They are the living parts of the coral. And everything that is darker are the dead parts of the coral. So the living part is always forming on top of the dead parts, and that's how they create this 3D structure. But if we move to unsaturated water, here we have an omega of 0.73. We still have corals living there. They have mechanisms to produce aragonite, even in unsaturated conditions, but we lost all the 3D structure because thermodynamically all these dead parts that were in the other reef, they dissolved. So they can still produce, and the organic parts will prevent the aragonite to dissolve when the living part, but all the dead parts don't have this organic kind of protection, and thermodynamically they start dissolving, and then we lose the 3D structure with all the consequences for the ecosystems. So that's why we wanted to study in this section here. So this is a track that is repeated every other year since 2002 from Lisbon to Greenland, and it's co-founded by Spain and Portugal. So 1 year is Spanish lead and the other year is French lead. And in here we study the circulation of water masses, the transformation, and also we monitor the carbonate system to see if we have changes in pH, changes in omega aragonite, etc. So during the next few slides, I'm going to talk about results of this section here. So the first is the water mass distribution, and again, scary equations here, don't worry. I just want to illustrate how we did this. So we evaluated the water mass distribution and the transformation of water masses using a technique that was developed in the 80s, which is known as optimum multiparameter analysis. It was really used at that time to understand how the circulation of the global ocean, where water masses were formed, and lately to understand a bit of the processes that were going on in the global ocean. And now it's more used to see distributions of parameters and sometimes understand the transport of some tracers. So what this analysis is based off is if we have a water sample that we take in a cruise, we can know where the origin of that water is. We can trace back where this water was last in contact with the atmosphere and tell this is coming from 50% from water from the Labrador Sea, 60% from water from Mediterranean basin, for example, right? So for doing that, we have to assume that what we see in our sample has only been subject to mixing. Nothing else happened to our properties. That's why we need conservative properties to do that. And that's why we have here potential temperature and salinity, as most of you may know. They are conservative parameters. And then we also add these parameters that maybe surprise you that we use them because they are not conservative. They are oxygen and nutrients, but we can make them conservative by adding this delta oxygen here, which accounts for the changes in oxygens that happen when we have the cycling of organic matter. So this OMP analysis can give us the distribution of water masses, but can also give us insights of the cycling of organic matter. And if we add another parameter, the alkalinity parameter I was telling before, we can also track the carbonate system in our mixing equations, right? So it can tell us lots of information, and I want you to keep these extra parameters in mind because almost at the end, I will go back to this for the Southern Ocean. So these were the results of the optimum multiparameter analysis, or OMP, for the period 2002 to 2010. From the section that you can see here, the one I was showing before, from Lisbon on the left to Greenland, on the right, to Greenland on the left. And another topography feature that I will make reference later is the Ibanez Ridge here. It's kind of this underwater mountain in the middle of the ocean, which is part of the Mid-Atlantic Ridge. And I will also talk later about the Erminger Basin, which is basically this basin over here, and the Iceland Basin, which will be from the Ibanez Ridge to the right up to this kind of a small mountain here. So just when I talk later, you know where I'm referring. But what I want to show you here is that if you remember some slides before, I was telling you how the Atlantic Meridional Libertarian circulation started and which water masses were for the starters and what was created. So we wanted to check if the OMP was able to reproduce what we know that is happening. And if you see here, we have close to the Iberian Peninsula, the central waters in surface. Suddenly when we go, in this case it seems west, but the section also goes north. So if we go north, we don't see anymore the central waters and they start to appear the subpolar mud waters. As you remember, the central waters are the precursors of the subpolar mud water. And at the end, we stop seeing again the subpolar mud water and we start seeing the Labrador Sea Water, which is this water here. And it's the most prominent water mass in this section. So this water, as I said, is formed in the Labrador Sea, and the variety that we see here is a variety that was formed in the 1990s. It was a big convection event that formed lots of water, and you see how it came from the Labrador Sea, which is here on the left, and it reached the Iberian Peninsula, and it spread all over the North Atlantic. The signal could be seen really far away from the Labrador Sea. And you can also see, as I was explaining, these deep waters, the overflow waters, the Denmark Strait overflow water, which is the densest water in the North Atlantic. And then we have the Iceland and Scotland overflow water, the one here, that comes on the right-hand side of the Reykjanes Ridge. So I don't know if you see it really, yes. So this is Iceland, so it comes south from Iceland on this side of the Reykjanes Ridge, and then south of the section it crosses the ridge and comes back north again. But you see it's not as dense as the Denmark Strait overflow water, and this gap here is what is taken by the Denmark Strait overflow water. We can also see the influence of the Mediterranean water, and see we are in Lisbon. It's kind of far away from the Mediterranean Sea, but we can still see the influence of the Mediterranean water here. And this is also another concept you have to take in mind, because this is part of my postdoctoral project here. And at the end, as we can also see the deep waters of the Northeast Atlantic deep water, which is this water mass that it was represented in the first diagram of the general circulation of the oceans. So this is the water mass that represents the 40% of the deep waters in the global ocean. And we don't only wanted to know where in the water column were these water masses, but we also wanted to know which water mass transform into another and how they recirculated here. And again, there's lots of numbers and things in this figure. If you are curious, I put the references to all the papers here. You can check them. But what we did here is to create a box between the A25 section, the one I was talking before, and the sections I was showing before, and the Greenland, Iceland, and Scotland seals here in the north. So we had like a closed volume, and then we divided this volume vertically between upper and lower limb of the Atlantic Meridional returning circulation, and then also east and west divided by this Reykjanes Ridge I was talking before. So this Reykjanes Ridge will be a barrier for the deep waters. They won't be able to cross, but it will be an open, as you see here in this scheme here, it will be an open border for the upper waters. They can freely cross the ridge because they are shallower enough to cross. So I won't go in detail on all these numbers here, but basically what we corroborate is that in this box, inside the box, we see how the central waters and the subantarctic intermediate water, which is a water mass I didn't talk, but it's also formed in the Labrador Sea, they contribute to the formation of the subpolar mud waters. We also saw how in this box, the waters of the East North Atlantic Basin, which is the basin on the right, the upper waters cross over the ridge to the Irminger Basin, and then in this box here, they cross from the upper limb of the Atlantic Meridional overturning circulation to the lower limb. So it was like a transformation of water masses happening in the Irminger Basin, cooling them down and mixing them to the bottom. So we have a transformation from upper and intermediate waters to deep waters in this basin here. And then they left the bottom basin to the south with this deep western boundary current I was talking before. And the last finding that I wanted to highlight here is, as I said, we assume that this Reykjanes Ridge was a barrier for waters from this basin to this basin in the bottom box, right? But we found out that it wasn't the case. Some parts of the Iceland and Scotland overflow water that flows from here south crossed before going out of the section or of our box. And we found there was another paper that at that time was talking about this, and since this paper that was in 2015, there have been lots of papers saying that this overflow water can cross before in really small reaches that are in the mid-Atlantic ridge, which has consequences for the circulation of water masses and ocean acidification, everything, right? So that was the beginning. We wanted to understand how the water masses were formed, which water masses lead to which other water masses. And then we also wanted to know if the different water masses have different acidification signals, if we can really see that the deep waters were recently ventilated, they have different acidification rates or the same. So that's why we did these two works here. I will focus on the work of the Irminger and Iceland Basin for my presentation, but I encourage you to go and check the work of my colleague Marcos Fontela, who did the same in the East North Atlantic Basin, this part here of our sections. But I wanted to focus on the Irminger and Iceland basins because these are the basins when we see the overflow waters, the relatively new waters arriving. To this part here, we have relatively old waters in the bottom, And we don't see the acidification signal in the bottom. So here you are seeing a section of this omega that I was saying before. If you forgot about it, I will recall what this means. It was the ratio between the concentration that we see of carbonate in situ and the saturation. If we were over 1, we are happy. If we are lower than 1, if we are coral, we will start suffering the consequences. So if you see here, naturally, we have a gradient from supersaturated waters to undersaturated waters as we move deep into the water column. That this is normal. This is not related to ocean acidification. This is thermodynamics. And if you see the deep waters in both basins, they are already undersaturated. You can also see here the limits of the water masses that we'll talk about later. So we have the subpolar mud water, the upper Labrador Sea Water, classical Labrador Sea Water, Iceland Scotland overflow water, and then Margaret State overflow water. So if you see these acronyms later, you know what it is. And again, this is the Erminger Basin. On the left, we have Greenland, this mountain here, Reykjanes Ridge, and on the right, we have the Iceland Basin. And in the bottom, we see the difference, the change in the saturation state of aragonite in these 27 years that we had our studies. You see in almost the entire water column, but a tiny bit in the surface, and this is because one cruise was in summer, the other cruise was in winter, and this has slightly changed because of that. But if we ignore the first 50 meters of the water column, we see there is a general decrease in the concentrations or saturations of aragonite. And how that translates into this aragonite saturation horizon, the depth at which omega is equal to 1. We see in 1991, omega aragonite saturation horizon was here, deeper than in 2018. And if we could zoom here, we could see that in the Erminger Basin, this change was 400 meters. So in 27 years, we have a solving of the aragonite saturation horizon on 400 meters. So imagine if you were a coral living here, you were really happy in 1991, and suddenly in 2018, you are in undersaturated waters. So most of the corals in this region, 80% of them, they live in kind of this area here. So they will be affected if this continues to fall in the future. So as I said, we wanted to see if these different water masses here had different rates of change in pH and in the saturation state of aragonite, this omega term. So again, I don't want you to see all the numbers here, but visually you can already see that all the lines in all the plots are similar. So each line is one water mass, from surface to bottom. They are kind of ordered because of the initial state already, right? And we see the rate is similar from the surface waters, which are the red ones, and also from the deep waters, which are in the top one in purple or in the bottom one in navy here. So we see changes in pH and omega that are similar in the entire water column, which is related to the fact that the deep waters in this region are really new. They may have 20 years, 25 years. So they are carrying this signal of the human impact in the ocean. And it is a change that is significant in all the water masses in both basins. And as you were seeing before this, the aragonite saturation horizon was raising, you see the difference. So this aragonite saturation horizon was soaring at 6 meters per year in the Iceland Basin, but at 34 meters per year in the Erminger Basin, which means every year this depth is 34 meters higher and higher, which is exposing, you only saw a section, but if we see the entire North Atlantic is meters and meters of hectares that are exposed to corrosive water every year. So this is really concerning. So we wanted to also, again, don't panic about all the bars and all the equations, but we wanted to know which were the causes of this change. Is it really the aragonite saturation changing because of one pH? I know so in pH, but we also did the same in pH. It is changing because we have more anthropogenic carbon, or is another cause happening here? And I won't focus on all these bars here, but just look at the gray bars, which is the observed change, and the red bars. There's already maybe popping in your eyes that red and orange bars are closer to the gray bars. So the red is the total dissolved fine organic carbon, as I was explaining in the beginning, is the carbon dioxide dissolved by carbonate and carbonate. And we divided this total dissolved fine organic carbon into the anthropogenic part and the natural part. The anthropogenic part is the orange bar. So you see most of the change that we see in the red bar is caused by the orange bar. And this anthropogenic carbon is the main cause of this decrease in the aragonite saturation state and also in pH. So we know it's not a natural cause, at least for this region. What is changing the saturation states of aragonite. And we wanted to know if we assume that the trend that we see in the period of 27 years continues into the future, when will we reach the aragonite saturation state of equal to 1? So kind of how do you call it this? Well, this point of no return, kind of a tipping point. Yeah, this tipping point that we cannot go back, right? So here you see that the graphs are in logarithmic scale, and it's because the carbonate system is not linear. So if we increase a concentration of carbon dioxide, it won't decrease linearly the aragonite because of all these reactions that we were seeing earlier. That's why we did these projections in logarithmic, but on the right-hand side and bottom, you can see the non-logarithmic scales. So you see here, this is when omega is equal to 1, and at which concentration of atmospheric carbon dioxide it will happen. And for the different layers that I was showing earlier, and in the two basins together. So what I want you to take away from this is that if we keep as is happening now, nothing changes in circulation, nothing changes in our emissions, the entire water column in these two basins, and in this section in particular, will be undersaturated with respect to aragonite when the water masses are in equilibrium with the atmosphere of its 860 ppm per volume. But the intermediate waters, the ones I was telling you where all 80% of the corals in the North Atlantic live, will reach undersaturation at 600 ppm per volume. This number will maybe not tell you anything, but this is slightly higher than the concentrations of carbon dioxide that will cause the 2-degree warming that we're talking about in the Paris Agreement and all that. So if we reach the 2-degree warming, we will be also endangering all the North Atlantic cold water corals as well. So then we stopped with this unhappy story, and then we moved south to the Southern Ocean. And this is a cruise we did in December 2019, just before pandemic, lucky us. And we wanted to, in this project, which is called Castar, that for British is really nice because that's how crema catalana is called in the UK. So Castar, it stands for Carbon Uptake and Seasonal Traits of Antarctic Remineralization Depth. And basically this project wanted to know, it's more based on the organic part of the carbon system, but they still wanted to know how deep this organic carbon reaches in the Southern Ocean. It is represented by this green kind of funnel figure here. So depending how deep this organic carbon reaches, it will be in a different water mass, and it will affect for how long this carbon is stored in the ocean. If you see here, it seems that it will reach really deep, it will be better, but if it reaches this acronym here, which means the circumpolar deep water, this water upwells back south of the polar front and will bring back this carbon again to surface relatively fast. But if it reaches the Antarctic bottom water, it will store the carbon for 1,000 years, for example, right? So we wanted to see if we could see that. That's why we went to do this section here, south of Chile. And we did a repeat of this 5 stations 4 times during the boreal bloom, and to see if we could see how biology is affecting, how deep the organic matter reaches, and if it really has any influence of this uptake of carbon by the ocean. But my part was not on the organic carbon, as you maybe already thought. I'm in the inorganic part. And we wanted to understand why we see this distribution of inorganic carbon in our section. And for those that are unfamiliar with the inorganic carbon or the carbon pumps, the concept, just had it here. So it's another concept that was created in the 80s. And basically, is any process that creates or maintains this gradient that we see here in the dissolved inorganic carbon, or in alkalinity, or in any parameter. In fact, almost all the tracers in the ocean have pumps. So if we won't have these pumps, we will have a DIC section that will be constant in the entire ocean. But these pumps create these gradients that we see. So the pumps are the soft tissue pump, the carbonate pump, and the gas exchange pump. These are for the inorganic carbon. So my study was focusing in kind of tearing apart the drivers of what we see, what is creating this distribution here. So we know part of it will be by mixing of different water masses coming from different parts with different signals. Part will be the soft tissue pump, part will be the carbonate pump, and part will be the gas exchange pump. And as if you remember from a few slides before, I was talking that this OMP analysis can tell us about the soft tissue pump, this delta oxygen, and if we add alkalinity to the system, it can tell us about the carbonate pump. So I won't show any more results because this is work in progress and this is going to be in YouTube. I will present later, maybe when we have the results finished. But we want to see if we can really see the influence of biology in at least the inorganic part of the carbonate system. And now moving to my Severo project, which is named MEDOC, which stands for Mediterranean Sea Natural Alkalinization, a mitigator for ocean acidification. And maybe you still remember about this alkalinity parameter. We know the Mediterranean Sea is a really important carbon sink because it has really alkaline water. So the more, if you don't recall, the more alkaline a water is, the easier it is for that water to take up carbon dioxide, and less changes in pH we will observe. So that is why Mediterranean Sea is really important carbon sink, even if it's a really, really small basin compared to the global ocean. And because of climate change and increases in evaporation, alkalinity will increase in Mediterranean Sea, so it will be prone to still take up more carbon dioxide. And as you see here in this graph here, we also have a formation of water masses. It's like seeing the entire ocean inside the Mediterranean. And because it's so small, this turnover happens in a human lifetime. So we can see the consequences of climate change really fast here. So that's why I wanted to see if these changes in alkalinity are really alleviating pH changes in the Mediterranean Sea. And if you remember in the sections before, we could see the influence of Mediterranean water in the North Atlantic. And there are lots of cold water corals in the Mediterranean Sea, but in the North Atlantic as well. And the idea is if we can see the changes in alkalinity here, how they affect the waters in the Mediterranean, and then follow this signal to the North Atlantic and see if that is projections into the future could kind of alleviate a bit what will happen related to ocean acidification. And why no one did this before is because there was no single database with all the data that was available for the carbonate system in the Mediterranean Sea. So luckily, we are collaborating with Marta Alvarez in EEO, she lead an effort called Carbonate, which is putting together all the black dots that you see here, which are cruises led by different institutions that happened since the 1979, to collate all of them and see if we can have a global picture of the carbonate system in the Mediterranean Sea, because so far we only have kind of pieces of each institution and each sub-basin of the Mediterranean Sea. And I'm also incorporating with help with Arturo that is there, trying to put together the two time series here from the ICM, also to study not only these cruises that maybe happens once a year, to study all these time series that we have, 4 time series, one here in Lebanon and these 3 here that are relatively long, and we can maybe see more seasonality and changes through that. And with this, I would be happy to take your questions. Thank you.
Thank you very much for this very interesting talk. Anybody has questions? Yeah.
Well, thank you for the amazing talk. It was very interesting. I have 2 questions. The first one is about if there's any estimate about how much carbon those cold water corals store, And my second question is to get your opinion about the suggestions of increasing the biological carbon pump through ocean fertilization and other methods, just to get your ideas about that.
Sorry, can you repeat the first question?
Yeah, how much carbon, if there is an estimate of how much carbon these cold water corals are storing?
Okay, so the problem with the cold water corals is they are really deep into the water column. So these estimates, maybe someone did a cruise sometime and they found there was a reef, and there was someone noted, oh, there is a reef here. So there is no, that I'm aware, an international survey of monitoring all the reefs. So this is a guy that really took all this together and make a database with all the records of people that did another survey in another, with another focus, and they discovered a cold water coral. So they are still not really studied and survey. So some people are starting now to focus on them, but there is, I don't have any answer for your question. And regarding the biological pump, so there are different mitigation strategies now being discussed to reduce the carbon dioxide concentration in the atmosphere. So there's no more in the ocean that this should be. But so far, none of them seem feasible to uptake as much as we need to take up. So they have been doing fertilization studies, enhances in alkalinity, but they are really local and just kind of pilot studies. I think what is more now being pushed is what is called carbon capture and storage, which is done by the same people that basically take up the oil from the reservoirs. They put it back, the carbon dioxide, back in the reservoir, and it can be under the sea or it can be on land. But it's still also pilot studies, and it is estimated that at the rate we are doing it now, it won't be enough either to mitigate our climate change. So I think there is a lot of discussion now because we see that we are going too close to this 2-degree warming, but I don't see any, so far, any solution for that. But thanks for the questions.
More questions?
I think Celia was before.
Thank you very much, Maribel. I enjoyed a lot the talk. Very clear. I was wondering that, but you were saying that the Mediterranean is it will be in nice systems to keep CO2 because of the alkalinity. But there is another thing that is against this, that is the temperature, no? Because it's a warmer water, even in deep waters, and the respiration can be higher, and maybe you should, but you will take this into account.
Yes, the idea is to do similar what I did with changes in omega, to see if we can link the changes in alkalinity to changes in temperature, and do this also this driver analysis at the end. But of course, I'm aware of the special conditions of the Mediterranean Sea, and also maybe we are seeing something now, and in the future, well, I know there has been a switch in the formation regions of some waters in Mediterranean. Maybe this happens again in the future because of increasing stratification, etc. So that we don't know yet. And of course, I'm not doing models. Everything is based on observations so far. Maybe in the future, I can collaborate with the modeling part of ICM to see if we can predict, if we move the formation of water masses, how that will affect. But the idea is to see if we can see changes in alkalinity and understand from where it comes from. Is it only because we have increase in evaporation, or is it because we have maybe more groundwater arriving to the different formation areas of the water masses? So that's the idea of the project.
Thanks also for the talk and the explanations and everything. There was a graph. I kept thinking about that all the time. You showed the change with the time of the Granite, not that one here, and it looks like for some areas there is actually an increase up to 2000 or so, and then a decrease.
Yeah. So you refer here.
Yes.
Yeah. So this is the subpolar mud waters, and these are the upper part of the water column in the North Atlantic, and it has been so because of the North Atlantic oscillation, you have an kind of shrinking, and so the subpolar gyre is smaller or bigger depending on the North Atlantic circulation. So the subtropical waters can arrive more north or south depending on the NAO, right? So these periods, this variability is linked to more subtropical waters with more salinity arriving to the region, and then the NAO changed, the subpolar gyre expanded again, and then we have more subpolar waters, and that's why we see this. So it's also linked to the salinity change.
Okay, do we know the time scale of that? Because maybe we are seeing like a snapshot of some cycle. That's,
yes, for the surface, the subpolar water, definitely we could have split it into periods because it's more affected by this more coming or subpolar mode waters or subpolar mode waters. But for the others, most of it is the signal is less clear in terms, well, maybe in pH, you can still see them.
Yeah.
That this Iceland basin is closer, if you remember the map, I don't know, it's closer. If you continue here to the south, this is the Iberian Peninsula somewhere here, so it's kind of closer to the subtropical gyre. So that is more affected by these fluctuations of how north or south this gyre reaches. But thank you.
So thank you, Maribel, for an amazing talk, but very, very clear, very well explained. Thank you. Still with this figure, so we don't see or you don't see a big difference in the slopes or in the trends, in the temporal trends. You say that this is because even the deep waters are quite new, quite recent. Still, shouldn't you see this 20 years delay or time lag between one water mass and the other? I mean, is that a convincing explanation for you that, okay, this is so new that we don't see the difference, or should we see them maybe with a longer time series, maybe with a higher resolution? So what's the limitation?
Yeah, so that's a good question. So here we have one repeat every other year, right? And it's only summer. So we don't have this temporal resolution of maybe this seasonality or signals that have lower frequencies. But and also something I haven't explained here, and it's a debate that is happening in the carbonate system community, is how we present this, because here we are presenting a pH, which is a logarithmic scale, and it's not telling you about the starting point kind of. So this water you see is, for example, here is 8/8, but the waters in the bottom are already below 8. So their change, it seems similar, but maybe if we do it in concentration of protons, it will be maybe different. So it's just because how the logarithmic scale transformed this into something comparable in a sense, because I mean, the person that created this pH concept was to have it easier, because if this is and I will have a really, really big number to put here, so that's the easier to represent it, but it takes out part of what the organisms see. They don't see pH, they see the concentrations of protons, right? So maybe we did the same in this concentration. Maybe we could see these things, but I think making it logarithmic takes part of that variability. But yeah, I expect they should have different mechanisms affecting, because they are forming different regions, and they are subject to different drivers. For example, the overflow waters are formed in the Arctic or Nordic seas, but then they overflow to the North Atlantic, and then they mix in that overflow. They mix with the subpolar waters, and I think that's part of why we see similar signals, because it seems that these are deep waters, and they are having always in the bottom, but the reach is 200 meters deep, right? So when they flow, they are encountering the subpolar water just when they are flowing, and they are incorporating that's my vision. They are incorporating already the signal of the subpolar waters when they are crossing the reach. So I think that's part of what they are so similar at the end.
Thank you. And as for the ecological / biogeochemical consequences of deep ocean acidification, you seem to focus on the cold corals, which is okay. I mean, you showed us that they are widespread and probably large reefs and so forth. Are there any other consequences you can tell us about? I mean, are there other organisms suffering from acidification, or either living there or sinking particles that can get more rapidly remineralized or dissolved or whatsoever because of acidification of and the deep
Yeah,
ocean?
so I focus on the cold water corals because they live in the bottom, and it seems that everyone forgets that the acidification is also happening in the deep waters, right? Everyone focuses on the surface waters. Of course, they are the ones that are taking now the carbon and seeing the changes faster, but in some regions, and in particular this one, everything is connected kind of, and we see the signal in the entire water column. There was kind of we selected the cold water corals to push kind of for the message, right? Because if we say, oh, there is the same ocean acidification signal in surface and bottom, and that's it, people will be, okay, and what? So that's why we saw that the corals were kind of starting to be recognized as a really hotspot of biodiversity, and we decided to kind of, even if my study is only focused on chemistry, kind of see what if this happens, right? But you have changes in everything related to pH, since the velocity of sound, that will affect all echolocalization, not only for animals, but also submarines are also studying ocean acidification because of how you send signals. It can affect the cycles of metals, so it can be that the metal forms are more toxic in certain pHs. So if you start switching, they will be more toxic for certain organisms. And the consequences are all interconnected, because at the end is if we're changing the pH or the air you are living in, right? So everyone living in that air is affected. So everyone living in the water will be affected, even if they don't have carbonated structures. I think there are studies also with sharks that their smell is changed because of the pH of the water. So it seems that everything will be affected in a way, or if they are not affected as an adult, they will be affected in the larvae stage, because most of the larvae are planktonic. And then all these things and everything also again linked to climate warming and changes in seasonality. So it's kind of a puzzle that we are still starting to understand. But yeah, it's consequences for everything, and I think it should be maybe more present in the discussions. I remember when I defended my PhD, it was 7 years ago, and my friends that are not scientists were really surprised that they didn't know about this, and scientists know about this since the 80s, right? They know something is happening, and you hear climate change, but there's not much talk about ocean acidification.
Questions? Somebody?
There's one someone at the back of the room.
Hola. Thank you, Maribel. I think I've learned a lot with your talk. And I have a question, just adapt a curiosity. When you mentioned that this known return point, when is supposed to be? I think I missed some portion.
Yeah, so I was just showing, you cannot say, it's 160 in a fixed number, so it's just a projection into the future of something that we see in 27 years, and we assume it's going to be the same in the future. We assume same circulation, same emissions, same uptake of the water masses. So it's kind of a back of the page calculation, right? But just to give an idea. Yeah. And we wanted to see if it was close, really close in terms of concentration. And by doing that in concentration, and not in time, we are not following any scenario of the IPCC, right? So it's just concentration. If we reach it tomorrow or if we reach it in 100 years, it's just based on concentration. So those are the numbers there at the bottom of the screen. But this is when the water should be in equilibrium with that. So maybe it reaches 160 ppm, but the intermediate waters haven't been in equilibrium with that. So it has to be that the water reaches the equilibrium with that concentration in the atmosphere.
Okay. And have you tried to see when it's going to be, like to try to,
yeah, so that's the lines here. In time, it depends on the scenario that we follow.
Yeah, exactly, in time.
Yeah, so in time it depends a lot. If we continue in the scenario that we're following us, that is the worst scenario, it will happen before the end of the century.
Okay. And this is maybe a little provocative, but I think how long is going to last the petrol and the oil in this world? think I Googled it, just like, and it seems that like the oil will last for maybe 50, 60 years. So in 50 years, 60 years, we will not have any more oil to burn.
Yeah, but in 50 years, you're already at the end of the century, kind of. So,
More or less, yeah. Yeah, no, but yeah, probably.
yeah.
Well, I just
Yeah, but thank you for that point.
welcome.
Celia, tu volies preguntar? Sí? No, no, fes pregunta.
But one comment that in surface, maybe the organisms are used to have these changes that you have here in this long period. In surface, in a bloom of phytoplankton and after a bloom, they see this difference. So I guess in surface more or microorganisms are familiar with these changes, but not for a long time, I guess. But I have a question, a curiosity, which is maybe not the point of the talk, but it's about the remineralization part of your OMP models, that for the variables that are not conservative, you put this delta because of
Oxygen, yeah.
remineralization, and you assume the remineralization is the same for all the masses, water masses?
Yeah, I know the problem with the refill ratios and not being constant over depth. This is one of the kind of assumptions that you have with this analysis. You assume so this is the refill ratio that is fixed for all the water masses, because when you give to the OMP, what you give, it's the properties that you observe in your samples and the properties that you tell these are my water masses. And the OMP doesn't know anything else. And it tries to find the combination of water masses that best solves what you see. So we could have in a way tell the OMP that if it's deeper than this, I don't know, 2,000 meters, the refill ratio shouldn't be this and should be this one, to know for the known changes in the refill ratios. So far, I'm not aware that anyone did it, because it's more like an estimation of it. But if we wanted to really go into detail, I guess, yes, we should change the refill ratio according to what is known in the region to really tell what is this organic part.
Okay, I have a last quick question. You mentioned about, I think it was 35 meters or something like this per year of shoaling in the Irvinger Basin. Is that like the Guinness record of what has been reported so far, or is
it is not, but because there's not that many people that do these studies with long enough series. But I know in the Pacific they have it higher, but it is not related to human, well, in a way, not human-derived CO2, but it's more linked to the aging of the water. So in the Pacific, I think it's the NOT time series. They see faster changes in aragonite, but it's because the waters are getting older, so therefore you have more remineralized carbon there that is also changing the saturation states. But in terms of only caused by anthropogenic signal, this is the largest that I'm aware of, yes.
in this place, this particular place, what is the speed? It's like...
I don't remember
40 or
the
50?
rate of the soiling, but I remember that the surface waters here in our basin are around 0053 or 0061. I think they were like 70 something. So it's slightly larger, but of course that translates into faster changes in the swelling depth, yes.
Thank you very much again for this great talk, and that's it.
Thank you.